Okay, so today we'll start excitable tissue. In excitable tissue, one is muscle, and second we are going to talk about nerve. Okay, so first we'll talk about nerve, then we'll talk about neuromuscular junction, and then we'll go to the muscles. So what is meant by nerve? In nerve, in very vague term. So here, origin of nerve is start with the neural cell, and that neural cell is called neurons. Okay, neurons, which is one of the most complex cell present in our body, and almost our human brain is made up of almost 10 raised to power 10 number of neurons, more than trillions of the neurons present in our body. फिर भी आप लोग उसमें से हार्डली कुछ एक या दो ही यूज करते हो. Please use little bit more neurons so that you can get better marks. Okay, and there are different shapes, size, and there are different characters are there. And this is one of just the basic structure. It's just to describe the common structure. It is not like the all neurons look like this. So this diagram is just a representative, but a different shape and different size, a different length of the neuron will be there. And basically neurons divided into three parts. One is a body, dendrites, and axon. Okay, so body is also called as a soma, or also called as a perikaryon. So carry means nucleus. So nucleus का आसपास का structure. That's why it is also called as a perikaryon. It is in brain. There are different shape of the soma you can see. Most common is like a star shape, also stellate shape. Some neurons also show pyramidal shape, fusiform shape, or sometimes circular shape of these neurons will be there, and it gives to various processes. So processes, very fine processes, multiple number of processes are given. They are called dendrites, and one long process they are supplied, and they are called axon. Then they bond with the cell membrane, and that is uh, similar to that of the other cell membrane, and in that they bound a fluid that is called cytoplasm, and in neuron it is called neuroplasm. Okay, and they contain a central nucleus which control the all activity, but one structure which differ from the other cells, that doesn't contain any centrosomes. If you know, in the 12th, the centrosomes are very much important for the cell division, and this centrosome is absent in the neurons. So well developed neurons are absent the cell division, so they cannot uh, reduplicate. So there is a cell division is not possible in the neurons, because with complexity they lost the ability to uh, develop cell division. Okay, so once the neurons damage in your brain, there is no chances to replace with the new neurons, because we had a fixed number of neurons just after birth. जो birth तक जितने neurons बन गए उतने बन गए, उसके बाद we had a fixed number of the neurons. It can decrease, but it cannot increase because of lack of centrosomes. It doesn't cannot multiply. Then apart from this, it contains all other cell organisms present in normal cells, like it contains mitochondria, it contains endoplasmic reticulum, it contains Golgi apparatus. Apart from this, it contains small vesicles which contain neurotransmitters, and those vesicles are called Nissel's bodies. Okay, Nissel is the scientist who described about these vesicles, and these Nissel's bodies are present, and these Nissel bodies stores the neurotransmitters. Okay, so these are Nissel bodies. These are usually basophilic, means they attract the bluish color, and the dark blue color will be there. And the Nissel's body are nothing but the fragments of the endoplasmic reticulum, and these usually contain neurotransmitter and other enzymes which is responsible for the maintenance of the functioning of the soma or neurons. And these Nissel's bodies are absent in axon also, and they're also absent in dendrites. So this doesn't contain these dendrites, and axon doesn't contain any Nissel bodies. So Nissel bodies are usually present in soma. And whenever the cell get fatigue, or whenever there is damage occur to the cell, these are the bodies which first disappear from the soma, and this mechanism is called chromatolysis. So this chromatolysis, which dissolving of the initial bodies or disintegration of the initial body, and it shows that the cell is exposed to either too much work, so it can get fatigue, or it exposed to the any dangerous thing like toxins or poisons or any direct injury to the cell. That time, this disintegration or chromatolysis occur. Then, apart from this, it also contains the microfilaments and microtubules. These are called neurofibrillae, and these neurofibrillae start disintegrated or degenerated in the old age, and that is responsible for the Alzheimer's disease. And these neurofibrillae disintegrate and forms into fragments of neurofibrillae, that is called neurofibrillae triangles. This you can see in the biopsy of the brain of Alzheimer's patients. Then these also contain pigments that is called neuromelanin, which is giving a black color pigment, and these are seen in various neurons like neurons of the substantia nigra, and these pigments are nothing but they're called lipofuscin, so it contains a little bit of lipid and black color pigments. Then come to the dendrites. Mm, Dendrites are small multiple process, more than hundreds of the processes are present on the body of the neurons, and they are usually receivers of the signal from the different cells. So they synapses with the different cells and receives the impulse. Once these dendrites receive signal, they generate the membrane potential, and mm, that carries to the soma and to the synapses, and they're responsible for the carrying signal from the different cell and receives, and once the signal analyze, and then it sends signals to the axon. So dendrite receive करता है, cell उसको analyze करता है, and then it sends the output through the axon. So axon is like a longer process of the nerve cells, and it arises from the one extension of the soma, and that is called axon hillock. So this is a axon hillock which doesn't contain any cell's body, and just after the axon hillock, which first portion of the uh, axon is formed, is called initial segment. And this is axon hillock is the site where actual action potential is generated. Neither soma will able to generate the action potential, neither dendrite will able to generate action potential, but only the axon hillock where the action potential is generated. And it is because the axon hillock has a contain higher number of voltage gated sodium channels, which are responsible for the action potential. These number are very less in other part of the body and dendrites. And the fluid which present in the axon are called exoplasm, and the cell membrane of the axon is called axolemma. So if you come to the end, the axon will multiply into divided into small multiple processes, and each process will have a little bit dilatation in the end, and these are called synaptic knobs. And these synaptic knobs are responsible for the synapses of the axon with the other neurons or other cells or other muscles. If you see this axon when they're moving out of the body, and once they come out of the CNS, they are supplied with the layering of the cells. And these are the Schwann cells, which forms a multiple layer surrounding, wrapping around the axon. And the sheath which formed by the Schwann cell are called myelin sheath. And these myelin sheaths are responsible for the insulation or faster conduction of the cell. That we'll see when myelinated and non-myelinated we'll see. Okay. So this myelin sheath, which is if you see, these are how the Schwann cell forms. So this is like a okay. So this is a okay. This is the axon, and this is a Schwann cell. So Schwann cell will make a multiple rotation. It will wrap like this continuously on the axon. So it will be something like this. And the cytoplasm which present in the Schwann cell will disperse, and it will only on the outer side. And only cell membrane will keep on attaching to each other firmly. So here it is like cell membrane firmly attached to each other in the center. and that usually made up of lipid bilayer, and they attach with each other, almost like they stitch each other, and they firmly attach and will form insulating covering on the layer, and that is called myelin sheath. With each Schwann cell will wrap there, and there is a gap between these two Schwann cell, and that gap of almost less than 1 micron will be there, and that gap will expose the neuron to the extracellular field, and that area where the gap is there is called node of Ranvier, and each branch will almost form the uh, covering goes up to uh, 1 millimeter distance. So 1 millimeter distance के बाद 1 micron का gap रहेगा, then again 1 millimeter distance, then again 1 micron gap will be there. So this node of Ranvier will be there. And now, what is the advantage of this myelin sheath? So myelin sheath will increase the speed of conduction. So what happen normally if there is no myelin sheath, the conduction action potential will generate like from this to this, and so at each every micron will be there is more, and action potential will keep has to generate it, so it will create a slow conduction. But in a no, myelinated fiber, so this is myelination, here is myelination, here is myelination, so action potential generate from node of Ranvier to node of Ranvier. So there is hardly it will take few time to generate, and so it will create a faster generation or faster now conduction. Apart from this, it insulating doesn't allow the spillage of the impulses from one neuron to the other neuron. So that is another advantage of myelin sheath. And third, if the injury occur to the axon, this myelin sheath will provide a structure to regenerate the nerve. So only regeneration occur only in myelinated neurons, and unmyelinated neurons, if they get damaged, the regeneration is not possible. Then come to the type of the neurons, depending upon the processes and all those things, like poles. Dendrite is one pole, axon is another pole. Depend upon the poles, these neurons has been divided into unipolar, bipolar, or multipolar. So unipolar, these are usually in the embryonic stage, where the only one process will be there. Then it divided into axon and dendrite. So this is one of the example of unipolar. Then another variety of unipolar is called pseudo unipolar. There is one process will come that actually look like it's coming from the one process, but there are two processes. One goes towards dendrites and one other form axons, and this is called pseudo-unipolar cell, and that is usually seen in the sensory neurons. Then come to the bipolar neurons, where the only one dendrite process will be there and one axon process will be there, and this type of the bipolar you will see in the ganglions like vestibular and cochlear ganglions. Also, nasal olfactory neurons are also having the bipolar neurons, and also bipolar cells also we are seeing in the retina. Then the most common type of the neuron is multipolar neurons. There multiple dendrite uh, will be there, and one axon will be there, and that type of the neurons are present almost everywhere. Those are multipolar neurons. Then depend upon the function of the neuron divided into motor neurons, which carries impulse from the brain to the rest of the body, or sensory neurons, which carries impulse from the rest of the body to the brains. Then depend upon the function of the neurons divided into different zones. So the first is receptor zone. These are usually dendrites, which receives the signal from the different part of the neurons, and with the receptive generation, and they generate uh, local potential, or also called postsynaptic potential. This postsynaptic potential will spread in the membrane of the soma, so that membrane of the soma will make the changes depend upon the dendrite receiving the impulse. Either it can be excitatory, so there is increase in the membrane potential, or it can be inhibitory, there will decrease in the membrane potential, and this will keep on submitting. So एक receptor को बहुत बार stimulate कर लिया, multiple receptors stimulate different type of stimulus, they will keep on accumulating and uh, accumulate together, submitted together, and will make a final change in the neuron. So, and it will make hmm, initiate the action potential in a initial segment and axon helix. That is called a zone of secretion, zone of transmission. So zone of all or none transmission. So either यहाँ पे action potential form होगा या नहीं होगा. If action potential form, this action potential conducted to the axon and goes to the terminal end, and where the neurotransmitter is released, and that create a zone of secretion. When neurotransmitter release and it combine with the other synapses, and it can increase or decrease the memory potential of subsequent synapses. Then come to the neuroglial cells. These are the supporting cells. So each cells almost hundreds of the glial cells will be present with respect to each neuron, and the different type of the neuroglial cells are present. One is called macroglia. It is one of the large cell. It is of ectodermal origin, and it is a star shape look like. That's why it is also called as astrocytes, and it again divided into protoplasmic astrocyte and fibrous astrocytes, and these are usually responsible for the myelination of the cells within the brain. Then come to the smaller type of cell, that is microglial cells, and these are different origin of these microglial cells. So this is one of the smallest cells, and you have multiple processes which bind to the blood vessels, and also it causes a responsible for the phagocytosis. These are microglial cells which are responsible for the phagocytosis, and what a neuron is damaged, the neuron will be removed, or if myelin sheath will be damaged, the myelin sheath will be removed, or other tissues will be damaged, that can be removed by these microglial cells. Okay. Then come to the structure of the nerve. Now is nothing but the bundle of axons, and that coming out of the CNS or going into the uh, CNS is formed by the nerve. Okay, can we take a 2 minutes break? So, structure of the nerve. Nerves are nothing but the bundle of the axons compacted together, which is coming outside the central nervous system. So, if you see the structure, the group of the neurons are combined together, and they are lined by the inner lamina of Schwann cell, and they stick to each other and to form a fasciculus. Okay, by the fasciculus, this is covered with the fasciculus, and this is called endonerium. So nerves are covered with the endonerium along the Schwann cell. The endonerium sticks to each other to form one fasciculus, which is covered by the another layer of fibrous tissue, that is perineurium. And perineurium will come together and form a bigger bundle that is called nerve. And this is a much tougher fibrous tissue along with the connective tissue. That tougher fibrous tissue, that is a epineurium. The epineurium fall together and forms a nerve.
बॉयज हॉस्टल लिखा है यहाँ तो हर हॉस्टल में यही हाल है हर हॉस्टल में ये तो होटल है इसलिए यहाँ पर
बड़े प्रॉब्लम है इन लोगों को हॉस्टल में. Okay, the main function of this epineurium, perineurium, or endoneurium is try to protect the nerve from the stretching or pressure or any other of the stress. So they prevent it from the fragile nerve is very fragile, from fragile of nerve to get a physically damage. So that is the main function of this epineurium, perineurium, and endoneurium. Then come to the exoplasmic transport. It is a conduction which occur actually. Axon not only transport the signals, but also axon provide the transport of the substances from the body to the synaptic nerve, and also from the synaptic nerve to the body through this exoplasm. This now is transport is occurring, that is called exoplasmic transport. So exoplasmic transport are two types. One is rapid transport, which goes from the body to the soma, and is use the microtubules, and this transport is occurring both direction. Anterograde from body to the soma and retrograde from the axon to the body. Okay, and the moment is anterograde somewhere around 100 to 400 per day. Well, retrograde is a little bit slower. That is somewhere around uh, 10 to 100 per day in retrograde flow. The slow transport, it is usually unidirectional, and it is from the body to the soma, and its transport is not through any microtubular. There is just a flow of the protein from the exoplasm, and these are usually larger fragments like a neurofilaments or tubulins. They are transmitted, and enzymes are transmitted. This slow transport, and this transport is just 0.1 to 2 per day from the soma to the postsynaptic knob. The advantage of why we this need this transport mechanism. Okay, why we need this transport mechanism? That these enzymes and microtubules and all this thing transported to repair or restructure and maintain the axon or activity of the axon. And this from this microtubule rapid transport is occurred. The neurotransmitter has been transmitted. What are synthesized in the uh, soma, it goes to the exoplasm, and also sometimes various organelles are also transmitted like a mitochondria and all those things, so that it will be provide energy to the axon terminals. Apart from this, retrograde transmission, the retrograde transmission will occur like viruses, toxins can transmit, and infection can goes from the nerve to the CNS. Then come to the properties of the nerve. It has one first property is excitability. So in excitability we can discuss like resting memory potential, action potential, and second important property is conductivity. So ability to transmit the signal from one cell to the other cell. So these are the two properties of the nerve fiber. Resting memory potential we already discussed. So here we're going to talk about the action potential. So action potential is defined as a self-propagated, spontaneous change in the memory potential when stimulated by the threshold stimulus for sufficient time. How we can define action potential? Self-propagated, spontaneous, non-decremental change in the membrane potential when stimulated by the threshold stimulus for sufficient time. So if we see the phases of the action potential, it starts with resting membrane potential, then we'll apply the stimulus. This is experiment. I mean, in our body will not doesn't get this stimulus artifact. This is an experimental. So experimental, when you stimulus that makes some changes, that stimulus artifact, then there is it will take a time to increase the memory potential. That time is called latent period, and then there is a increase in memory potential. So it increase in memory potential, it reaches to the firing level. So in neurons, in case of neuron, firing level is minus 55 millivolt. So once it goes to the firing level, there is rapid change in the memory potential, that is called overshoot, and it goes beyond the zero and it reaches to the peak, that is somewhere around plus 32 plus 14, that is called overshoot. That is called spike potential. Sorry, not overshoot, that is called spike potential. Once it reaches to spike potential, then it comes down slowly, and this phase is called repolarization. And once it reaches to the resting memory potential, it goes below the resting memory potential, and that is called after hyperpolarization. So these are the phases of the memory potential. Resting memory potential is minus 70 millivolt, which we already discussed in detail about resting memory potential in the previous class. So we are not going to talk about this in much detail. Then stimulus artifact, it is due to leakage of the current when we stimulate the system, that is a nerve muscle preparation. That time it affect the sensor, and that creates a stimulator artifact. Latent period is occur because of stimulus when you stimulate the nerve to reach the nerve from the stimulating electrode to the impulse to the recording electrode. वहां तक जाने के लिए जो time लगता है, that distance which travel, that time is one of the time which is responsible for latent period. And second, to generate the action potential, it takes some time, and that because of that there is a latent period. Then come to firing level. Firing level, when we stimulate the electric current, it opens the sodium channels and slowly will raise the membrane potential, and it slowly pile up, and it will reach to the level at minus 55 millivolt, which is a critical membrane potential, where the voltage gated sodium channels are sensitive to that minus 55 millivolt. Once we reach the minus 55 millivolt, it opens almost all voltage gated sodium channels present in the neurons, and that will causes rapid entry of the sodium inside the cell, and that will lead to overshoots, and it goes increase the membrane potential, and it goes beyond the zero potential, and that is called overshoot, and it can reach up to the reach to the plus 32 plus 40 millivolt, and that is called spike potential. Because of this rapid rise and there is a rapid fall, so there is a one pointed end will be there, the spike like of the structure will be created, and that's why it is called spike potential. Once it start coming down, there is a slow repolarization will occur, and that because of fall in the membrane potential, and it is because of opening of sodium channels. Sodium ions start going out of the cell, and that causes fall in the potential. But as the sodium channels are very slow to act, slow to open and slow to close, even if they reach to the resting membrane potential, they remain open, and so there are more sodium channels open, and membrane potential goes below minus 70. There's somewhere minus 72 millivolt, and this is called after hyperpolarization. Now come to the ionic basis, which already little bit we talked. Resting membrane pressure is because of unequal distribution of the sodium and potassium ion across the membrane. Okay, then we'll start about a depolarization phase or action potential. So this phase is called depolarization phase. So this phase is depolarization phase. So when we stimulate and apply the electric current, that will opens the sodium channels and increase permeability of the sodium channel. So sodium channel will start entering into the cell, and membrane potential from minus 70 start increasing as the sodium channels entering. It start increasing, and once it reaches to the firing level, it opens the M type of the sodium channels, or also called M gates. These are very sensitive to the voltage membrane potential, and when minus 55 millivolt, that rapidly opens all these voltage gated sodium channels, and this causes rapid entry of the sodium ion. So sodium ion enters rapidly inside the cell, and because of rapid entry of the sodium ion, there is a rapid rise in the membrane potential. Now, once it goes above the plus 30 millivolt, then it will stop much entry of the membrane potential, and there are reasons for that. As the membrane potential become positive side above the zero, it inactivate the sodium channel. So there is an inactivation of sodium channel, so sodium channels take time to inactivate, and there is no more movement of the sodium ions through the sodium channel. At the same time, it also start opening the voltage gated potassium channels, and as the voltage gated potassium channels open, this causes exit of the potassium ions. So there is late loss of the positive ions from inside the cell, and that will limit the height of the action potential. Now, how the repolarization occur? As there is no more sodium channels are opening, so there is no entry of the sodium ions, so there is no rise in the membrane potential. But at the same time, there is opening of the potassium channels, and now the potassium ions start moving out of the cell, and that will causes a fall in the membrane potential. So if you see the blue line, it shows the movement of the sodium ion, and this green line shows the movement of the potassium ions. And as it going down and down, more and more potassium ion will open, and it will start rapid falling in the membrane potential. But as the membrane potential goes below minus 40 millivolt, it start closing the membrane potassium ions. But potassium ions slow to close, so it continue to fall even after it reached the membrane potential. So there's a moment of the potassium ion continue to go, and that's why it goes beyond the level of the normal resting membrane potential and become more negative side, and that is called hyperpolarization. And this mechanism is called hyperpolarization. So there are two channels are involved: voltage gated sodium channels, voltage gated potassium channels. Voltage gated sodium channels opens, तो potassium sodium enter हो जाएगा, and that will increase in membrane potential. Voltage gated potassium channels open होंगे तो potassium cell से बाहर होंगे, and that will decrease membrane potential. Now here I use word channels. So if you know where we used to membrane transport mechanisms, अगर आपको याद है, तो channels word has been used for facilitated diffusion.
Facilitated diffusion.
So these are the examples of facilitated diffusion. So sodium channel, potassium channel, जो action potential में जो भी होता है, all these are occur through the passive transport mechanism. Okay. Now there sometimes question comes, अगर action potential sodium अंदर आ रहा सेल के पोटेशियम बाहर जा रहा है, तो does it create a disturbance in extracellular intracellular fluid concentration? और कुछ देर के बाद तो ऐसा हो जाना चाहिए था कि पूरा सेल के अंदर पूरा सोडियम होना चाहिए था, सेल के बाहर पूरा पोटेशियम होना चाहिए. That doesn't happen. So there are two factors. So whatever changes occur in this concentration of the sodium is only occur close to the cell membrane. पूरे साइटोप्लाज्म में सोडियम का concentration नहीं बढ़ता. The changes occur near to the cell membrane. So पूरी सेल है, axon है, तो this changes of the sodium concentration occur only close to the cell membrane, outside also and inside also. And second thing, whatever the sodium enter inside the cell and potassium moves outside the cell will be rapidly corrected during this resting phase by the sodium potassium ATPase. Now, to measure the characteristics of the excitability, one of the feature is strength duration curve. This is experiment done to see how excitable tissue is. And here what we do, we'll slowly keep on increasing the strength of the stimulus, and it will stimulate the tissue till it shows the response. The one point will come, if keep on stimulating, there will be no response, and one point will come where that this is the strength, suppose above the 10 millivolt. That one point will come when you stimulate for long enough that can initiate the response. So the minimum strength of the stimulus or minimum intensity of the stimulus, when applied for adequate time, that can initiate the response is called rheobase, or we can call it as a threshold stimulus. So this is a threshold stimulus. Rheobase is nothing but the threshold stimulus. Okay. So this will give you idea about the excitability, and time required to initiate the action potential is called utilization time. Now what we will do, we'll increase the strength of stimulus and we'll make it double the rheobase. And then we'll apply the stimulus on the muscle and we'll note the time required to initiate the action potential in the now. Okay, so we'll double the strength of the stimulus. So कुछ 11 के 12 के ऊपर था, तो अभी 24 के आसपास किया, and then we see the time, and now time required to initiate the stimulus, that time is called chronaxie. Okay, and chronaxie is a good indicator of the excitability of the tissue. Longer the chronaxie, less is the excitability. Smaller the chronaxie, better is the excitability. And now here you see there are two graphs has been plotted, A graph and B graph. A graph is for the now and B graph is for the muscle. So if you see now muscle, now is much excitable as compared to muscle. Then come to the second property is all or none law. I guess our time is over. So I will share this PPT, and if you had any time, we can continue this discussion later. Okay?
Okay, sir. Okay.
Okay, sir.
Bye. Have a nice day.